So next going to section 13.1. So what do we know in the photosynthesis previously? That's chapter 5th in class 10th. So there are some experiments to understand the photosynthesis process. The first experiment was done with the starch and iodine reaction. That's called a starch iodine test. So this experiment was initially done to verify whether chlorophyll or green part is essential for photosynthesis or not. But we already know that the green part has the chlorophyll, so which absorbs the light energy and that's where the photosynthesis happens. But earlier in 17th century they don't know anything. So they wanted to check whether chlorophyll, the greener part in the leaf, is really required for photosynthesis. So what they done, they've taken a plant with a variegated leaf. So variegated leaf means a leaf which is having different colors. So green, the whatever you see, the shade, the colored with the some part of the leaf is greener, some part of the leaf is white, some part of the leaf is yellow. So that's why it's called variegated leaf, like a mosaic leaf. So the greener part has a chlorophyll in that. So other part doesn't have chlorophyll in that. So only the greener part has chlorophyll in that. So what they have done, they have taken the variegated plant and they kept in the dark to remove all the starch which is produced through photosynthesis previously. To remove it from the plant, they kept the plant in the dark condition for 3 days. So there is a zero starch now. So then later they have shifted it to sunlight. In the sunlight, after keeping in sunlight for several hours, they took the leaf from that plant and they put it in boiling water and they also put it in alcohol. So why they have done this? Just to dissolve chlorophyll. They want to do the staining with iodine. So chlorophyll is already a greener pigment. So you can't see the iodine pigmentation properly. That's why they wanted to remove all the chlorophyll pigment. So the experiment is already over. That's why they want to remove all the chlorophyll pigment. So when this leaf is treated with iodine, so iodine in normal is orange to brown in color. But when this iodine is treated with the plant, the leaf is treated with iodine. So the iodine reacts with the starch, and only the areas where the green pa- part was there in the leaf were turned into deep blue. How it turned into deep blue? So iodine reacts with starch. The starch has a amylose sugar and amylopectin sugar. So amylose has a helical structure. So in this helical structure, the iodine gets binded and it's give blue color. So wherever the green part is there, green region is there, there it has the blue staining color, deep blue staining color. That means wherever the green region is there, there only the starch has been produced. So starch is produced, that's why it's reacted with iodine, it's giving deep blue color. So iodine is at normal as orange brown color. When it reacts with starch, it produces deep blue color. So what it indicates, only the region where the green color, the chlorophyll or green pigment is there, that part only is producing starch. So starch is produced through photosynthesis. So that's why it indicates chlorophyll is essential or green part is essential for photosynthesis. This is proven by starch iodine test. So another test is KOH experiment, potassium hydroxide experiment. This experiment is done to check whether CO2 is essential for photosynthesis or not. So we don't know, earlier they don't know anything about photosynthesis. They wanted to check what is really required for photosynthesis, whether CO2 is required, oxygen is required, whatever. And so this test was done to whether CO2 is required for photosynthesis or not. So they have taken two plants, plant 1 and plant 2 with leaves, and the same thing, they are kept in dark room to remove all the starch. And again, after keeping in dark room for 3 days, why they want to the keeping in dark room? So they want to make sure that there is no previous starch left. So they are making an experiment. So that's why they are making it zero. So they are making zero starch, and later one plant is kept in a closed jar, another plant is also kept in a closed jar, but plant 1 inside the closed jar they have kept KOH substance, KOH chemical. So what KOH chemical does? KOH is a basic substance. It absorbs or it reacts with acidic, that's the CO2, because inside the air is there. So normally air has a CO2 and oxygen and everything. The CO2 is there. This KOH absorbs all the CO2 and it make potassium carbonate and water. So it's a basic and carbon dioxide is acidic. So basic acidic reaction. So it absorbs all the carbon dioxide and the carbon dioxide is not available to the plants. Here there is no KOH, so carbon dioxide available to the plant. So it's a tightly sealed. This air packed sealed. There is no air going. There is no air coming from outside. So whatever the air is inside, only that much is available for the plant. But here the KOH is kept. All the CO2 is taken down by the KOH. So again, when they are kept in the sunlight, and again when they are treated with iodine test, only the plant 2 showed starch. That's a deep blue color. Why? Because there is a CO2 is available here. But plant 1 there was no starch and there is no blue color. Why? The CO2 is absorbed by KOH. That's why plant can't use CO2 to produce carbohydrates or the starch. That's why it proves that the carbon dioxide is required for starch formation. The starch formation happens through photosynthesis. Thank you.